Okay, hello everybody, welcome back, and I'm delighted to introduce our next presenter, Dr. Euan Ashley from Stanford University. Before I introduce Euan, just a few housekeeping notes. I'm Kevin Davis, first of all, the publisher of CNEN, Chemical and Engineering News. We are delighted to be the official media partner with LabRoots for this inaugural precision medicine event, and to have shared in the responsibility for putting this fantastic agenda together. A couple of housekeeping notes, as I said, we won't be able to accept any live questions today because for scheduling reasons we're having to pre-record this a couple of days early. We're very grateful that Euan was able to squeeze us in between frenetic grant funding deadlines and a trip back home to his native United Kingdom. So we're grateful that we're able to snag him for 30 or 35 minutes to record this, but that will preclude any live Q&A, unfortunately. As far as the actual presentation goes, you can enlarge the slide window on your screen by clicking the screen icon in the lower right-hand corner of the window. If any technical problems should arise viewing or hearing the presentation, then please click on the support button on the top right-hand corner of your screen, or you can submit your problem through the Q&A button, which you'll find lower left. This is an educational presentation, and we're offering free continuing education credits. So at the conclusion of Euan's presentation, if you click on the CE button, which you'll find bottom left-hand corner of the web page, you can follow the instructions there to claim your CE credits. So now it gives me a great pleasure to welcome Euan Ashley, who's recording this from the UK. Euan is an associate professor of medicine, genetics, and by courtesy pathology at the Stanford University Medical Center. As you will quickly surmise, Euan is a native of Scotland. He studied at the University of Glasgow, completed his medical residency and a PhD at Oxford, before moving to Stanford University, where he specializes in cardiovascular medicine and genetics. His group is focused on precision medicine. He's been in this field for some time. In 2010, he was one of the lead authors in a study in the Lancet on the first clinical interpretation of a human genome, a landmark paper in the field, and he now routinely applies genome sequencing approaches to the diagnosis of patients in their network. He's also a co-founder of Personalis, a genome-scale diagnostics company. Euan has given himself plenty of wiggle room with a title of a talk that is simply Towards Precision Medicine. So Euan, take it away.
Well, thanks for the introduction, Kevin, and it's really a pleasure to be here and participating in this very unique webinar series on precision medicine. I'm really delighted to talk to you today and just lay out some of the exciting new advancements that are bringing us, I think, closer to a world of much more targeted medicines for our patients. Here, first of all, is my disclosure slide, and I am founder and advisor to Personalis, and as you'll see on the bottom, increasingly talking to technology companies. It's interesting how in this new world the technology companies are increasingly interested in medicine. Well, this has become the meme for our community over the last year, since President Obama in January 2015 at his State of the Union said this: "I want the country that eliminated polio and mapped the human genome to lead a new era of medicine, one that delivers the right treatment at the right time." And that was his launch of the Precision Medicine Initiative that he laid out in more detail at a press release, just a press event, just a few weeks following this announcement. And it was really an exciting time. It was really the starter pistol for a race towards a new beginning in thinking about how we do this. And over the course of the next 40 minutes or so, I'm going to lay out some of our thoughts on where we are along that road. Of course, for President Obama, this wasn't a new thing. In fact, as a senator from Illinois back in 2006, when he had a little bit less gray hair than he currently has, he had a bill that essentially laid out many of the plans that later came to be part of the Precision Medicine Initiative. Of course, this bill didn't make it out of committee, but it shows the long-term view that President Obama had over the course of the last decade and his commitment really to making this happen. And of course, a couple of papers here from Francis Collins and one from myself, just laying out really what we think about the exciting time that is ahead and thinking about what might be here in this new era. Well, part of the Precision Medicine Initiative is the formation of a Precision Medicine Initiative cohort, the idea of bringing together 1 million Americans who would participate in pushing the boundaries of science by volunteering and sharing their own data for the betterment of others and for the discovery of new therapies. And a number of different themes had really enabled the idea of this million-person cohort. It was something that, of course, President Obama had envisaged really early on, but also Francis Collins, the current director of the NIH, had many years before talked about the benefits of population studies. And in particular, the timing is right now for such a cohort because of many of the things shown here on the slide. In particular, I think we're all very familiar with the precipitous drop in the cost of genome sequencing. But of course, we're also entering an era now of participant empowerment, and it's long delayed and should have come some time ago, but it's happy now to see that it's really picking up power, picking up momentum as we start to put the power back into the hands of the participant. Subjects in research studies are no longer. We are looking for partners who want to learn with us and really participate by contributing their own data, but also their time and energy towards doing things better. Of course, a major enabler of that idea is the mobile health revolution and the idea of sensors that are embedded in phones or in wearables that are really, again, putting the power back in the hands of the participant. And I'll tell you a little bit about some of our own experience in relation to that over the course of the next little while. And finally, we are starting to think about data integration. The major change that happened over the last 5 or 10 years in our healthcare system in the United States was towards electronic healthcare records. And of course, they're in the most early days and are often based on pretty old style technology. But we're beginning to think about getting the data out of the back ends of these electronic medical records and integrating them with some of the more modern approaches that are laid out above. So really the time is right. So what do we mean by precision medicine? And here's how the definition that I'll use over the course of the next little while: to target or prevent disease by measuring it more precisely. And the example that the President used in the State of the Union was cystic fibrosis. And this has become really the poster child in many ways for this approach. And the idea is to use technology, and in this case genomic technology, to improve the subclassification of a disease or condition so that it can be targeted more precisely. Well, in this case, cystic fibrosis really was thought of as a single condition. It's autosomal recessive, affects probably 70,000 people worldwide. And it is caused by mutations in the CFTR gene, the cystic fibrosis transmembrane conductance regulator. And mutations in that gene and in that protein can lead to different functional effects. And some of those, for example, in the earliest study, the G551D mutation leads to a reduction in the opening probability of the channels. And these are shown here in the picture. But of course, in this particular setting, the channels are reaching the surface. So let's imagine that you had a drug, and ivacaftor is one, that could increase the opening probability of the channels at the surface. If you had a drug like that, then it would be very useful to use it if you had a patient whose channels were making it to the surface but weren't opening enough. However, if you had another patient, for example, one of the 85% who have the F delta 508 lesion, then those channels are often destroyed because of misfolding and don't make it to the surface. And in that case, it doesn't matter how good your drug is at improving the opening probability of channels on the surface, it isn't going to be helpful for those patients. And this is really the essence of precision medicine, that by understanding the disease better at a molecular level, we can target it better with appropriate treatments. And in fact, another drug that improves trafficking for those class 2 patients, lumacaftor, has now been approved, and even this combination strategy is possible for the F delta 508 patients whose channels reach the surface, they sometimes have a gating abnormality that can also be helped by ivacaftor. So this really describes a new world. Another important area is precision oncology. And really, cancer has become redefined as a genetic disease. It has for many years. We've appreciated that somatic changes can predispose to cancer, and really started to define cancer biology in a very different way. We do have FDA-approved drugs for EGFR, an ALK rearrangement, lung adenocarcinoma. But what we find is when targeting those, often targeting the driver mutations, that we find that quite often further somatic mutations led to therapeutic escape. And so these therapies were not as successful as we hoped. In the new era of precision oncology, I think we find many, many areas where genomics can help, both with diagnosis and with therapy. We can think about germline risk, first of all, by DNA sequencing. We think about identifying driver mutations, but we can also think about neoantigens. We know that tumors are a nidus for the immune system, and that there are challenges with checkpoint receptors and the autoinhibitory effect on T cells. But we can sequence T cells, and we can sequence the T cell repertoire in the new era. We can use RNA sequencing to identify the checkpoint receptor and ligand gene expression in the tumor. So our understanding of the biology of cancer has changed greatly. And when we think about vaccination as a strategy in cancer, and the combination of that sort of strategy with the power of genomics, we really are describing a new era and one of great excitement for the future of patients with these conditions. Well, from the future, I'm going to take you a little back into the past, because it wasn't so long ago that genomes really cost a huge amount of money, more than we could ever have thought of spending on our patients. Back in 2009, one of my colleagues, Steve Quake, had sequenced his genome with just a couple of people in his lab for $40,000, which, well, that seems like a lot today, but at the time it was a tenfold reduction from the genome prior to that. And it occurred to us at that point that perhaps we should start thinking about what would happen if patients were much more routinely going to walk into their doctor's office with their genome, if you like, on a stick, or certainly on a hard drive. And so that led us to start to think about what sort of algorithms would you need, what sort of tools would you need, how much computing power and manual power would you need to analyze a genome. And so we put together a team back then. My group was focused on Mendelian disease, and we started to think about genome-wide disruption of the open reading frame, starting to think about how you would build some tools to do that. This was a helicose genome, so even an earlier time before Illumina genomes were the standard approach. We also knew that pharmacogenomics was going to be important, and my colleague Rosalban has been running for many years with Terry Klein, the PharmGKB tool. And so we had a database that was already very well curated and available to tell us about the effect of genomic variants on drugs. And finally, we were increasingly improving the GWAS catalog, and my colleague Atul Butte had put together a database for common variants and their effect on disease. And we thought that combining these different approaches and tools would allow us perhaps to start to provide a personalized approach to medication treatment and to diagnosis of disease and thinking about future risk. And we worked that up over the course of the latter part of 2009. One of the stories that came out when the paper was published was this one around trying to give personalized recommendation for statin therapy. Steve Quake himself was a 40-year-old man at some risk for coronary artery disease from his family history. But the guidelines shown in very small print here were really no help, because when you plug Steve into these guidelines, it said at the end, physician choice was the best thing. So that wasn't very helpful. And so we wondered if his genome could be more helpful, and what we found were that there were rare variants in his genome that would suggest he had indeed inherited the family history that he was able to tell us about. There were common variants that pushed, hard to tell how much, but that pushed a little bit in the direction of positive risk of disease. But I think much more importantly, we were able to find he had variants that showed that he would likely respond to the drug. And even one further than that, he also had an absence of the variant that would suggest he would likely have a side effect. And so we were, for the first time, able to give a personalized recommendation, really from a person's whole genome sequence towards their medical care. And this was certainly inspiring to us, but it took many, many months and took a lot of manual labor. And over the course of the last few years, we've tried to work on both reducing that amount of time and also thinking about improving our algorithms. Well, one of the groups that contacted us shortly after we published that paper was this family, John West and his family. He had become famous as the CEO of Celexa, who managed to sell all of his core business to Illumina, and had been one of the first families ever to be sequenced on the Illumina platform. And so he wondered if we could help in the same way we had done with Steve's genome to interpret his family's genome. And we took a deep breath and went ahead with that. It was obviously exciting to have a whole family and to be able to think about doing analyses like the one you see here, an inheritance state analysis across family quartet, and thinking about exactly which parts of the genome were inherited to which child from which parent. And of course that is important because we can start to work out once again which part of the family history is most relevant, and did the individual kids inherit the actual risk for disease. And in John's case, he was particularly focused on his risk for pulmonary embolism or for clotting, because he had suffered a pulmonary embolism, taken warfarin as a good patient should, but then suffered another one. And so he had been particularly curious to work out if there was anything in his genome that would help him give his children advice on how to avoid a similar thing. And we were able to provide some help with that and make some recommendations on that basis. And the power of then having a whole family of genomes was obvious to us, as well as thinking about the benefit in having parents when you're trying to work out what, for example, is de novo. Because many of our conditions, and we think of congenital heart disease as one in that category, but in many cases we see a child who's affected with parents who are unaffected, and our mind goes to compound heterozygosity or de novo models of inheritance when we see that. And so this was one of the first times I think we were able to think through what sort of algorithms would need to be in place to think about inheritance across a family when we think about the clinical part of their genomic analysis. We also were able to reduce error by using the family information to reduce or to narrow in the range in which we could define crossovers below 1 kb for the first time. And phasing is important. I mentioned compound heterozygosity. Well, being able to do chromosome length phasing is something that is possible using the computational framework shown here, but is otherwise challenging. As we move towards longer reads, of course, we will much more commonly be able to phase genomes, but that world is just coming up. Well, I mentioned the reduction in cost in sequencing, and that's certainly been transformative. The change from a human genome project funded at $3 billion down to a $1,000 genome has been quite spectacular. But of course, the cost is only one thing, and there are some situations in medicine where you really want it to be fast as well as cheap. And one of those situations is the intensive care unit, and in particular, the neonatal intensive care unit. And so I wanted to tell you this story about one of our patients who came into the neonatal intensive care unit at Stanford Children's Hospital. And this little baby suffered from long QT syndrome. So I may take just a moment to explain what that is for you. An ECG is shown here on the left, the gentleman with a number of pads stuck to his chest. It's an electrical tracing of the heart. And you end up seeing waves that are, for historical reasons, labeled P, Q, R, S, and T. And that is the depolarization of the heart, followed by the repolarization of the heart, so the recovery each beat. And the distance between the Q and the T wave is known as the QT interval. And if you have an inherited condition of the heart muscle called long QT syndrome, then that distance is long and you are predisposed to dangerous rhythms. And that's what this little baby suffered. In fact, she was brought in at 36 weeks of gestation and delivered by cesarean section, and the first day of her life, she suffered 5 cardiac arrests. It was a very dramatic presentation. In fact, she became one of the youngest babies ever to have an implanted defibrillator. You can see it on the right of the screen there in her abdomen with the lead tunneled up towards her heart. And as I mentioned, the long QT interval itself is not so much the problem as the predisposition to very dangerous rhythms like the one shown on the bottom left of the screen, which is torsade de pointe, which is turning around the points, a very dangerous heart rhythm where the heart essentially pumps out no blood. And so this little baby suffered 5 cardiac arrests in the first day. But long QT syndrome is redefined in the new era, in the precision medicine era, as a genetic disease, and one where we have specific therapies that work better for specific subtypes of the disease. And in this case, if we had a way of knowing which subtype this little girl was suffering from, we could personalize the therapy. But the current test costs $5,000. It sequences 5 genes, and it usually takes about 6 weeks to come back. So we decided that wasn't good enough, and we made it for a new route. We spoke to our friends at Illumina, who agreed to do this rapid turnaround genome sequencing over the course of a weekend. We drew the blood on a Friday, and by Monday afternoon, my postdoc James Priest had worked through the genome through one of the rapid approach algorithms to find this KCNH2 variant that was a known cause of long QT. And because we were able to work out that the little baby was suffering from the form of long QT syndrome caused by a potassium channel gene rather than a sodium channel gene, we were able to personalize the therapy of this little baby. And I'm happy to tell you that over the course of several months, she has done very well, left the hospital. She still has her defibrillator in her abdomen, but this is a picture of her here, and she still continues to do very well to this day. So I think that's a nice example of precision medicine in action. And the idea that the transformative potential of genomic technology being available, not just cheap, but fast, is really quite significant. Well, another part of this enabling theme of the precision medicine cohort is the idea of participant empowerment, and the idea that patients are no longer waiting for the doctor to tell them what should happen or to wait for the doctor to go find the next breakthrough, but are rather doing that themselves. And so this is the story from Matt Might of hunting down his son's killer. And this is an actual blog post that he wrote. I found my son's killer. It took 3 years, but we did it. Not quite like this. I should clarify one point. My son is very much alive. Yet my wife Christina and I have been found responsible for his death. My son Bertrand has a new genetic disorder. So the story that took Matt to the point of writing this blog post was that his son had an undiagnosed disease that was affecting his neurological development. And he had managed to have access to an early exome program at Duke, and some variants had been found. But the challenge is that if you have a disease that no one has suffered from before, and a variant is found in a gene that has never been associated with the condition before, then it's very challenging to prove causality. And Matt had been well known for a blog that had talked about surviving his PhD, and so he went to the internet to try to track down other patients that might have similar symptoms, including some rare symptoms like a lack of tears. And he wrote this particular blog post that became viral and was ricocheted around the world. And it led, in fact, to the finding of several other individuals with the condition and the narrowing down of the cause. It was featured in this New Yorker story. Here's Bertrand on his bed. And it led to this paper in the end, describing that mutations in NGLY1 and this new condition of NGLY1 cause an inherited disorder of the endoplasmic reticulum-associated degradation pathway, glycanase being the enzyme, and the pathology being consistent with this reduction in the degradation of these particular molecules. But the most important thing about the story is really what happened next, because this was an important landmark. But really, Matt and other parents like Matt Wilsey, shown here, patient at Stanford, really started to get together, to work together, to find other families, to connect, and to start to raise money so that they could fund researchers to try to find a cure. And this really describes a new world, a brand new approach where the participants are the patients and the families, and they're really pushing the research forward as hard as anyone else. And I think new technology is what facilitates that. The idea that you can go on the internet and you can write a blog post and you can connect with other individuals. And I think our community is catching up. We're now thinking about ideas like Matchmaker Exchange, connecting individuals, physicians, and scientists across the world who might have patients with similar symptoms, or you might have found a gene that you believe might be causal, but you need to find that second case. And this is really describing a new world. In fact, the new world was illustrated as much as anything by the fact that this particular editorial was written by the parents themselves. Excuse me. These approaches are one of the many inspirations for the Undiagnosed Diseases Network. The Undiagnosed Diseases Program has been very successful and has been formed at the NIH medical program over the course of the last few years, but was expanded just in the last 2 years to a network around the country. And now you can go to a centralized gateway and apply. And again, it's the patient who applies. Excuse me. If you're a patient with an undiagnosed disease, you can gather your records together, get a letter from your physician, and apply directly to the gateway, where your case will be triaged to one of the different sites who will review it, and you can have a relationship with that site as you visit. It's really an opportunity to apply the best of technology, whether it's genome sequencing, whether it's multi-omic, excuse me. The Undiagnosed Disease Network is an opportunity to apply the very best that medicine has to offer to these very hardest of cases, whether it's genome sequencing, whether it's omic assessment of the immune system. The network tries to give hope where there has in the past only been stagnation. Going even beyond the genome then, we can think of ways in which technology can really facilitate precision medicine. And in particular, you might be familiar with this, which is the back of an Apple Watch. This particular one is gold-plated and $17,000, and so it's quite possible that is not the one on your wrist right now, but it's certainly a new world where we're able to have timepieces that are able to shine LED lights into our skin and tell us about our physiology. Talking of that, here's one from Samsung. This one is not commercially available, but has multiple channels of LED light. It's also possible that it can take an ECG that I described earlier, an electrical tracing of your heart, by placing your right thumb on your left wrist. We have been testing these devices in our group because we're very interested in the potential impact on patient care. But as with genomic approaches, we're very interested in high accuracy, and we need to know that these devices operate at clinical grade before we can start to use them. And so we've been testing them all in the lab. Here's the initial test from the Samsung device as compared with the polar heart rate monitor that goes around the chest. And here's some data from the test that we've done looking at sitting, walking, running, and cycling in the lab. There's also a field-based component to this particular test, where we send people out around Stanford campus, cycling, and having their cardio-metabolic parameters measured by this rather attractive blue mask that you can see shown in the picture. And because you have two arms and you can fit two watches on each wrist, we are testing multiple devices at once, and you can see some of the earliest data from our testing here. What we found is really that the heart rate monitors of the latest generation are significantly improved on the generation before, and are under many circumstances quite reliable. Not surprisingly, perhaps cycling is a little bit more reliable for a watch-based sensor because your wrist is a lot more stationary on a bike than it is while running or even walking. But these are certainly significantly better than they were. At the current level, I think we would advise caution in over-interpreting the caloric estimates because they seem to be continually still very noisy. But the heart rate monitoring is significantly better than previous. And we got particularly interested in this because we had the opportunity to work along with Apple and several other academic centers in launching the ResearchKit platform on iOS. And we were focused on cardiovascular health. And we worked along with the American Heart Association. This was the study that we put together, which uses surveys based through the phone and physical activity monitoring and a fitness test through the phone. And it was launched at the Apple Spring Forward event in March of last year. One of the really, I think, transformative things about mobile health and about the opportunity to build ResearchKit is that we were able to get IRB-approved consent, informed consent, on a mobile device for the first time. And really, I think we were very proud of this as much as anything else that we did, to the ability to offer patients and participants information that was in a handy format on their phone. Each screen looks like this. There's an opportunity with a a simple sentence to learn more on each screen, but a key number of screens take you through and take you towards the end, where at the very end of after signing the consent form, you get presented with this screen. And this, I think, is even more transformative, because not only are you in charge in the sense that you're doing your own consent in your own time on the phone, you then are able to say exactly which data points you want to be part of the study. And if you get up tomorrow and decide that you don't want your date of birth to be in the study anymore, then you could pull out your phone, pull up the screen, and slide the slider back across. I think the ability to put the power in the hands of the participant is really transformative, and something we were very proud to be part of in pushing forward this new era. It's also something I think we'd be excited to see very much as part of the precision medicine cohort study. Another thing we'd be excited to see there is the ability to use security features that are built into the phone. For example, Touch ID. This has been developed and keeps all your personal information, perhaps your banking information and other information on your phone secure. So it should also keep the data you have chosen to share with a study secure. And so we were happy to be able to build that in. Also, we can use wearables. Here's my wrist and my Apple Watch reminding me to complete the My Heart Count survey. And by the way, if you're interested in this particular study, you can download it free from the App Store and join in. We currently are in the final stages of the first 7 days of collection. We ask every participant to give us 7 days of data, and then we leave you alone for a few months. But the second version of the study is just coming, where we start to randomize among different coaching approaches to improving and increasing your physical activity. Well, when Apple announces something, the world pays attention, and we went from really literally nobody in the study to 10,000 people in the first week, to 30,000 people in the first month, and to over 50,000 people now in the study just a few months later. This was really an unbelievable change when you consider the amount of effort that would have been required to collect this much data on 50,000 people previously, for example, 7 days of physical activity. How do we do that currently? We ask people, what did you do last Monday? How many flights of stairs did you climb last Tuesday? Do you remember? I can barely remember what I had for breakfast this morning, never mind what I did last Tuesday. So having a phone in your pocket, or a wearable that is monitoring not just your activity, but for example your heart rate, is really a new world from the point of view of doing these kinds of studies. Having it available on 50,000 people is quite significant. This was from our first data freeze, physical activity on 18,000 people, just checking to make sure that we have sanity here. This is hour of the day along the x-axis, and proportion of individuals active or inactive shown in green. And as you can see, it has the correct pattern. People get up in the morning, they exercise a little bit more at lunchtime, a little bit more at the end of the day. Of course, we're really at no more than 20% of the population are active at any one time. And so we have quite a bit of work to do, because we know that physical activity is among the most potent drugs that we have, and that it helps, that physical activity helps the health of every single system in our body. We can also use some of the approaches to high throughput data that have been pioneered. Machine learning approaches, for example, here just k-means clustering of the patterns of activity. Instead of us defining the patterns of activity, we let our participants define them by their own activity. And so rather than us deciding that one group is in one area and one group is in another, we can see what people actually do and let the data speak to that. And although this is an observational study, of course there's no causality here. We're already seeing associations between physical activity clusters and the presence or absence of disease. We could do fitness tests as well. This is how we would currently do a 6-minute walk fitness test in the hospital. There would be a nurse, a clipboard, and someone walking along a line. This is how we do it now, outside, with an iPhone in your hand, walking and turning around if you need to, but certainly in your own time on when it's convenient for you. If you wanted to look in the past and see for the 6-minute walk what would be normal, you might look to the literature, and you'd find these kind of studies, the largest study being 900 participants. And if you think of running 900 participants through a 6-minute walk, that is a long time it would take. In the first few weeks of our study, we had 5,000 people complete the My Heart Count 6-minute walk. And one of the joys of being able to do this is that we can return the result back to the individuals. In this case, we can show the cohort variation for the 6-minute walk distance and show where you as an individual fall as particular incentive to continue to be physically active and do more. And if you needed any more incentive, then the statistic from a paper that was published just a couple of years ago should be enough. We often think we don't have time to exercise, but the data for exercise and physical activity improving your life is really beyond repute. And in particular, 1 minute of exercise buys you 5 minutes of extra life. So you really do not have an excuse not, as soon as this talk is finished, to go out and exercise a little bit more. Well, and finally, for the last few minutes, I wanted to talk to you about data integration. We've talked about different data streams that are coming from high technology in the precision medicine era. We talked about genomic data and its size. We've talked about wearable data and its size. We haven't talked much about electronic medical record data, but we've mentioned that in the new era, increasingly we get information about our patients and information for participants in studies coming from that way. What we're interested in is whether we can combine this data, and certainly one of the ambitions of the Precision Medicine Cohort study is to be able to combine that sort of data, perhaps also with social media data, so that we can learn more about participant behavior. From the point of view of a physician, we would love to be able to integrate this data in real time at the bedside or in the outpatient clinic so that it's available for us to make decisions for our patients. And so we've been working along with the SAP Innovation Lab here at Stanford in developing a kind of next generation medical record, something where we could combine genomic data with wearable data and electronic medical record data. And this is shown right here. This is from a bioRxiv study that just posted last week. And here in this situation, we have 38 patients in an example clinic, all of whom have had their whole genome sequenced. And you can pick on one individual, as we did there, and look through. And what that system would do is look into their electronic medical record and do natural language parsing to essentially pull out ontologies or pull out particular events that would be associated with diagnoses. And you would see one here in red, shown with the number 1 at the top of the screen. We have a timeline series of analysis of the medical record. But at that point where there was one in red, a diagnosis was made. In this case, it was cardiomyopathy, a disease of the heart muscle. So that triggered a lookup for the VCF file for the genome for this patient for any genes that might be associated with cardiomyopathy. And you can see that they're shown there on screen just underneath. Other things we can do for every patient, of course, ABO blood type, the MHC, and thinking about HLA typing, as well as ancestry. The amazing thing really about the system, though, is it runs in real time. So as the screen refreshes, the natural language parsing is carried out on the data file, and the genome data is looked up in real time and brought to the screen in front of the physician. Well, we also have wearable data, and this is really just a pilot study, but of course, if you have heart rate and accelerometer for your patients, you can tell when they're exercising, so that's good, and there's a point in the middle of the screen there where the heart rate shown in blue and the energy in yellow are both high, and that would suggest the patient's exercising. But a little further along where you see the red bar, you can see the heart rate is high, but yet the yellows show that the energy level is low. And this is a point where the heart rate is high, but the patient isn't moving. So that would suggest a time of stress. And so we can use the wearables now to point out these things and bring that to the attention of the physician and bring it to the attention of the participant or the patient so that they can alter their lifestyle in a way that would lead to a reduction of their risk. Well, we're not quite at this point yet. You can't list your iPhone as your primary care physician. Well, some of my patients would like to do that, I think. And maybe they would prefer an iPhone as a cardiologist as well. But we're not quite there yet, but we're moving certainly in that direction. And I think this new world of precision medicine is an exciting one to be moving towards. I presented work from a lot of different people. These are a few of the folks from my lab and the groups who funded the work over the last few years that I've shared with you today. Here are the team from the My Heart Counts app. If you're interested in downloading it and joining the study, please do. We should have an Android version also coming out soon. I'll finish with a quick plug for our conference, Big Data in Biomedicine. If you want more of this kind of information and some of these speakers that you see shown on screen, come join us at sunny Stanford campus in May, and we'd be happy to welcome you where we can talk more about big data in biomedicine. The future then, I think, is bright. I think precision medicine is really a transformative idea. It's one for which the time is right, for which when we have genomic information really at our fingertips, when electronic medical record data is coming, and increasingly we can look to wearable technology to tell us much more and give us a much deeper picture about the physiology of our patients. Thank you for joining me today, and I'll be happy to take a couple of questions. So in terms of those questions, I have a couple here. The first question is this. It said, you were involved in the first in-depth clinical interpretation of an individual genome in 2010. How has that process improved in the past 5 or 6 years? And what do you consider the remaining chief problem areas? Well, I think that's a very good question, because I think that we've made a lot of progress in the last few years, but that we're not there yet. At that time, I think we had a group of maybe 25 people working for 6 months on one genome. And so we've definitely moved further ahead than that. We have much improved algorithms. Many parts of that process that were quite manual at the time are no longer manual. But the final mile, the final part, is still very much manual, because it means we go over individual new or well-known variants and look at the evidence for their causality. And in many cases, we still cannot do better than looking at the raw data in IGV on the screen. I think one by one we're getting better, but there are still many areas where improvements in the algorithms could really take us forward. For example, we're pretty good at single nucleotide variation. A recent study that we published just a year or two in JAMA showed that if you compare 2 different platforms for whole genome sequencing, then most of the time, a high 99% of the time, those 2 platforms would agree. But that figure dropped to around 56% when we thought about insertions and deletions. And if you think about the propensity for insertions and deletions, small indels, to disrupt the open reading frame and therefore potentially cause disease, then that's concerning. And if you think also then that structural variants, where we disrupt the reading frame even more, where we're equally poor at looking at those, then I think there is still quite some way to go before we have everything at clinical grade. We also want to think about coverage. If you can't see an area of the genome, you can't call it. And you need to be able to see enough bases at high enough quality to know what the variant you'd like to call there is. So I think we have some way to go. I think we've done a lot. We've made a lot of discoveries using the algorithms developed predominantly for cohort variant discovery. But to take this up to clinical grade will require a little bit more attention. And I think there was one more question mentioning that I'm a co-founder of Personalis, which is a genome scale diagnostics company. And the question is, how is Personalis making an impact in precision medicine? Well, again, I think this is a company that was founded with the idea that we have to bring genome sequencing up to clinical grade, and the idea of accuracy at its very core. And the company has focused a lot on Mendelian disease up to this point, and in thinking about the point I was just making about coverage and the capture approaches that we use for exome sequencing have large holes in them, sometimes even whole exons that are missing. And so the idea of augmenting that coverage and making sure that we really can make a call at every coding base position is really important, and one that Personalis has focused on. I think the idea of being able to replace current technology is based around the idea of non-inferiority. You have to at the very least be non-inferior to current technology. And if we think about what we use clinically, we use Sanger sequencing, and we use microarray technology to look for structural variation. So one of the interesting approaches Personalis has been taking is the so-called thin whole genome. So along with an augmented exome, there can be low coverage of the whole genome. This maximizes cost effectiveness, but also allows structural variant calling really in one single test. So that's a very exciting new area, I think, that Personalis is following. And I think the final one is that the oncology precision oncology is an area I mentioned at the beginning, and one that I think many are very excited by. The idea that we can have near-term therapeutics that will be personalized, whether it's through vaccination and other approaches. And so that's been another area of focus for Personalis. Okay. So thank you, in summary then, thank you for joining me